Assalamu alaikum. Eukaryotes. Eukaryote term comes from Greek, where eu means good or true, and karyote means nut or nucleus. So the cells with a true nucleus. We talked about prokaryotes and said that they are unicellular organisms, meaning that an organism is a single cell. As for multicellular organisms, including of course every organism we see with our eyes, they are all eukaryotes, and their cells are eukaryotic. Eukaryotes are divided into four categories: human and animal cells, plant cells, fungi, and protozoans. In this video, we'll talk about the human cells. In an organism, not every cell is the same, but all cells have common basic properties, which we will talk about in this lesson. The cell contains one membrane, made of a phospholipid bilayer. The head part is the negatively charged phosphate group, which is hydrophilic, and the tail part contains fatty acid, which is hydrophobic, based on so that the two tails face each other, and the negative phosphate is the one that reacts with water from both sides. The cell has a nucleus, which is the brain because it is the basis of the cell. For example, when the cell divides, the nucleus divides, and the daughter cell that has a copy of the nucleus can grow into a normal cell and it will have organelles. The nucleus is covered with a nuclear membrane or nuclear envelope, which is a double-layered membrane, outer and inner membrane, each of which is similar in structure to the cell membrane, with some additional features of its own. The nuclear envelope has nuclear pores, which are small pores that allow certain compounds to pass through the envelope. The nucleus contains all the genetic or hereditary material of the cell in the form of DNA chromosomes. The region inside the nucleus is called the nucleoplasm. The regions of the cytoplasm and nucleoplasm together make up the protoplasm. Proto means the first. That is the first thing that was created in the cell, because when the cell is formed or produced by cell division, the nucleus does not have an envelope, so the space in the cell is the protoplasm. Inside the nucleus, there is a small region called the nucleolus, which is responsible for the synthesis of ribosomes, which exit the nucleus via the nuclear pores. Then they divide into two groups: ribosomes attached to the endoplasmic reticulum and ribosomes floating around in the cytoplasm. And we will talk about ribosomes in detail in future lessons. Now let's move on to the organelles. To clarify, the organelle means the small organ. As the lung and the heart are organs in the human body, similarly, the organelles are organs of the cell, and they are essential for the function of the cell. The organelles are enveloped by a membrane similar in structure to the cell membrane. The endoplasmic reticulum is an organelle consisting of a network of sheaths folded together, and between these sheaths there are cavities. The endoplasmic reticulum is divided into two parts: the one proximal to the nucleus, which is the rough part, and the other one is the smooth part. Rough endoplasmic reticulum. The reason for calling it rough is because it contains ribosomes like granules that make it rough. We sometimes refer to it as granular endoplasmic reticulum instead of rough. The outer membrane of the nucleus is part of the rough section of the endoplasmic reticulum. That is, the nuclear membrane is continuous with the rough endoplasmic reticulum. In the process of protein synthesis, its genetic material is transcribed in the nucleus and then sent to the outside, so the endoplasmic reticulum receives the genes that came out of the nucleus and the ribosomes translate them into proteins. Then the endoplasmic reticulum does its special job, which is to check and verify the protein. Is it suitable for the required function or not? If yes, it is good to go. But if not, the endoplasmic reticulum will do some modifications to the protein, and this function is called post-translational modification. So if the modification was successful, it is good to go. But if the modification failed, then it orders a command to destroy this protein. This is the function of the rough endoplasmic reticulum. As for the smooth endoplasmic reticulum, it is smooth and does not contain ribosome granules, and its function is in the metabolism of carbohydrates and steroids. It contains also detoxifying enzymes that deactivate toxic substances by adding a hydroxyl group to them, so they become hydrophilic and the body can excrete them. It also takes a role in storing calcium ions, which makes it very important in muscle cells, because muscles need calcium for contraction. Let's move on to the next organelle, the Golgi apparatus. This is the cell's post office, because it sends proteins and lipids out of the cell. To understand its function, let's first go back to the rough endoplasmic reticulum. After modifying the protein, it will release the proteins necessary for the cell. But if it is a protein that is needed for a function outside the cell, then it will send it in vesicles to the post center, the Golgi, which will make the last modifications to the molecule and then sends it outside the cell with exocytotic vesicles. This is the function of the Golgi apparatus. Both the endoplasmic reticulum and the Golgi apparatus contain bulging domains in their structure laterally, which we call cisterna. The structure of the Golgi is like sheaths in the form of arcs that represent a hyperbola. The cis phase is the end that receives the vesicles from the endoplasmic reticulum, and the trans phase is the end from which the molecules are sent outside the cell. The mitochondria is an organelle enveloped by two outer membranes. Its main function is to produce energy. It produces energy in the form of ATP molecules, and it needs oxygen for this process. We usually use the term mitochondria, which is the plural form of mitochondrion. This is because they are usually present in large numbers in the cell, as one mitochondrion is not enough to produce energy for the cell. These were the main organelles in the eukaryotic cell. There are also other organelles, such as the lysosome, which destroy foreign substances such as viruses, the peroxisome, which breaks down toxic peroxides such as oxygen monoxide, and the proteasome, which destroys or degrades the damaged proteins in the cell, and the misfolded proteins that were commanded to be destroyed by the rough endoplasmic reticulum. Note how the organelle names end with the suffix -zome. This suffix usually indicates an organelle. All these organelles stand on the cytoskeleton, and the cytoskeleton is made of molecules that form tight networks inside the cell, to give the cell its shape and for the organelles to stand on it, and for the vesicles to move through it. Briefly, everything inside the cell is connected to the cytoskeleton, and we will cover more information about the cytoskeleton in upcoming lessons in this series. Now that we took an idea about the eukaryotic cell, its components, and how it works, we see that it is more like a small factory. The cell membrane plays the role of the security guard, as it allows the entry of certain substances only. The cytoskeleton is like the columns and walls that this factory stands on. The nucleus plays the role of general manager, as it is the basis of the cell and it synthesizes all the necessary proteins. The endoplasmic reticulum plays the role of quality manager, as it checks the quality of the protein. The ribosomes on its surface play the role of workers, as they translate to the proteins. The Golgi apparatus plays the role of a shipping manager, as it organizes each protein in a specific vesicle. And of course, the mitochondria are electricity, and oxygen is the fuel. This concludes our discussion about the subject. If you want to test your knowledge by solving the questions, continue the video. We said that everything in the cell is connected to the cytoskeleton, which gives the cell its shape, and we compared it to the columns and walls in the factory. The smooth endoplasmic reticulum takes a function in steroid synthesis, detoxification, and calcium storage. But checking the quality of proteins is the rough endoplasmic reticulum's job. Ribosomes are synthesized in the nucleus. Then they exit the nucleus through the pores that are in the nuclear envelope. Some attach to the endoplasmic reticulum and other float in the cell. Their function is to translate genes into proteins.